So genotyping and sequencing are different ways to move from biological specimens, spit or blood, to bits of information you can analyze on a computer.
Genotyping looks for a specific set of known variants, while whole genome sequencing looks for all the variants in your genome.
Both of which are becoming commodity businesses. What's new and interesting and exciting, difficult as well, is annotation,
which is converting A's, T's, C's, and G's into meaningful data about biological relevance.
Let's say you went through a private enterprise to be genotyped or sequenced.
Your data, either that set of specific variants or a set of all your variants, is sitting either in a cloud or in a hopefully secure data warehouse.
The first thing a company might do is compare your variants to a pre-existing database which tracks the correlations between phenotype and genotype.
You might be wondering, how is that database created? Well, it's created by compiling scientific literature which reports the correlation between variants and diseases or traits.
And our confidence in those correlations depends on a whole host of factors, such as the size of the study, the effect size of the variance, the p-value. Essentially, we're trying to achieve a statistically significant result.
A company's confidence in a specific correlation might be conveyed to you on some sort of scale, like a star system. As more and more studies confirm that correlation, you'll see more stars.
Let's talk about a different problem in annotation. What if variants, which don't have an overwhelming number of scientific studies or reports about them?
Which will happen a lot in whole genome sequencing.
So the first thing a company might do is just try to place those variants on particular genes.
And then try to find out what those genes do?
Absolutely. And then also trying to find out if the changes in the code actually affect the way your body produces proteins.
And that's where we don't have a lot of information to go on.
And now I'll refer back to the caveat we started with for whole genome sequencing. You might recall we said that whole genome sequencing would identify all the possible mutations that you have,
while genotyping will probably miss some.
But the drawback to that is it'll identify mutations for which we don't have much information. We don't know if they're functionally relevant. There's a lot of darkness there.
At the moment, genotyping can cost anything from $0, meaning your insurance covers it, to $200. But whole genome sequencing is still in the thousands.
So maybe there's a strong economical case for now to go with genotyping for carrier testing.
But in the future, as the price point drops, that decision might become more and more riddled with trade-offs.
We look forward to discussing those trade-offs with you then.